Hi, I'm Miren, and I'm a battlefield cell.
Hello, I am Jackie and I am a choke cell culture.
Hi, I'm Claudio, and I am a neutrophil.
Hi, I'm Crystal, and I'm a keratinocyte.
Tissue culture is used in biological research, in which cells are transferred to an artificial environment in which they can continue to survive and function.
Although it may sound similar, tissue culture is not the same as cell culture. Here, a small fragment is used to produce a whole plant. It is used for large-scale multiplication and improvement of crops. Animal cells must be supplied with fundamental amino acids. To grow, they require a surface that mimics the interaction they have with the extracellular matrix in an organism.
Now let's go back to the past, to the early developments of cell culture, where the fundamental techniques of cell culture started. Sidney Ringer developed a solution that preceded culture medium. It resembled bodily fluids and consisted of variable amounts of inorganic salts, glucose, stable pH, and osmotic pressure. These factors were modified according to the tissue and kept cells alive for short periods. Rose G. Harrison monitored nerve fibers of a frog growing for several weeks in fresh lymph fluids from an adult frog. Why is this important? To the development of in vitro and in vivo techniques and new opportunities for new study techniques. Montrose T. Burroughs found that lymph was not favorable for warm blood animals and used plasma instead. Jolene P. M. Bogelor and Eleanor Ehrlichman add hormones and plasma to Ringer's Baker's medium that was developed. It contained vitamins and blood plasma. Alexis Carrel, who won a Nobel Prize, contributed to the tissue culture by developing a prototype of the cell culture flask that nowadays is used worldwide. Immortal cells proliferate indefinitely due to the mutation. With this, Wilton R. Early, along with other researchers, discovered that proliferation from a single cell is possible. Immortal cell lines are often used in research in place of primary cells. They offer several advantages, such as they are cost-effective, easy to use, provide an unlimited supply of material, and bypass ethical concerns associated with the use of animal and human tissue. George O. Gay and his coworkers created an immortal cell line from cervical cancer tissue from Henrietta Lacks, who died of cancer that same year. It is the oldest and most commonly used cell line. However, there are different cell lines that are used on the present time, such as SF9 insect epithelial cell, Chinese hamster ovary cell, NCI60 NCI human tumor cell lines, HEC293 human embryonic kidney-derived epithelial cells, and number 1 HeLa cells.
And now, onto the reactants. The medium can be biological or of synthetic origin. The chosen medium should contain the necessary nutrients to allow the proper propagation of cells. The popular medium include MEM, containing only vitamins and non-essential amino acids, DMEM, which is sustainable for high-density cultures. It also includes phenyl red, a pH indicator and a light protector for the cells, and RPMI 1640, suitable for most type of mammalian cells. And remember, cells grow either in a single layer in a plastic surface or as a suspension in a liquid medium. As for the most important equipment, we have a biosafety cabinet. It minimizes the cross-contamination and creates a sterile environment. A contamination may kill the desired cell lines or enhance the growth of undesired cells. Incubators, where the selected cell will grow. Centrifuges, in order to separate the cells from the old medium. And finally, a cell counter. This will facilitate the control of a culture and indicate whether the chosen cell is growing or not. The general protocols include the initiation stage, in which the cells are introduced to the new growing environment, and the multiplication stage. This increases the number of cells in our medium. As growing cells leave less nutrients and secondary metabolites are created, this generates a less ideal environment, but this can be fixed by generating subcultures after 80% confluency from the original one. Once enough cells have been cultured, whichever experimentation was intended or any further step required for this may be done. For example, epidermal cells can be cultured and used in a variety of manners. On day 0, you should dip the biopsy sample rapidly in ethanol to avoid future contamination, then rinse thoroughly. On day 1, prepare the sterile material. Transfer the tube content into a petri dish and mechanically separate the epidermis from the dermis. Transfer the epidermal sheet into a tube and incubate at 37 degrees. Transfer the tube content into a petri dish. On a second petri dish filled with medium, dip the dermal part and mechanically extract HDMES. Transfer it to a tube containing medium. Incubate and filter the suspension. On days 2 to 4, for the epidermal cells, collect the supernatant and rinse. Centrifuge and resuspend the pallet. For the dermal cells, add fresh medium. Collect the supernatant, centrifuge, and resuspend before utilizing it.
The first application is focused on studying age-related diseases and their possible treatments. Cell culture is also really useful in the study of mutations and cancer because it is an alternative to subjecting living animals to carcinogens such as UV light, asbestos, arsenic, etc. When it comes to drug development, cells are more accessible for testing new compounds than if they were in the organism itself. It is the first step in drug discovery in many cases. Recently, cell culture has had numerous applications in vaccine production because traditionally, viral vaccines were produced in chicken eggs, which are economic but inefficient. Although not cheap, cells can be cultured in large scale and some viruses grow better in them. Furthermore, egg allergies can also be avoided. Fun fact, the Oxford AstraZeneca vaccine for COVID-19 relies on cell cultures produced in large bioreactors. As you might remember, rDNA technology is the introduction of foreign genes into cells for their expression. Although simple proteins can be produced in bacteria in this way, proteins that require post-translational modifications and complex folding need to be made in mammal cells. Fun fact, remember Chinese hamster ovary cells? Well, more than 60% of recombinant proteins are produced in these cells because they can grow in suspension. They express high amounts of protein and they have less risks of propagating human viruses. However, cell culture is not perfect and it has some limitations. Since growth mediums are rich in nutrients, unwanted viruses, microorganisms, or even other cells can grow if there is cross-contamination. In organisms, cells live in a complex environment. Therefore, cells in vitro do not grow as fast, and if the conditions are not replicated well enough, they might not grow at all. Because of this, many cells only last 30 to 50 doublings before they run out or start losing their differentiated characteristics. This leads to a morphology change. As you can see, cell culture has many requirements, so the amount of cells that are obtained from it is not that high.
Cell culture can be used to study and treat affected skin areas, like skin ulcers, which are open ones that develop due to an injury, poor circulation, or pressure. Also skin burns, caused by heat, radiation, electricity, or chemicals. And skin cancer. In this case, normal cells can be converted into cancer cells by using radiation and chemicals to understand the causes of cancer and develop drugs to treat them. Skin plays an important role in our body. It ensures homeostasis, and it is the first barrier that protects us from aggressive agents in the environment. Oh no, John got skin burned due to an accident, but we can help him by generating new tissue through cell culture. If we don't, these injuries can lead to circulation failure, problems with temperature regulation, sensory perception, infections, and even death. Let's not waste time and get to work. Before solving this case, it is important to know skin's anatomy. In the epidermal layer, we have main cell components, like keratinocytes, which are involved in cell growth and have tight intercellular junctions that prevent water loss. We also have the dermis, which mainly provides blood supply and delivery of nutrients. It contains fibroblasts, which form the extracellular matrix and collagen. The extracellular matrix is important because it is involved in wound healing and scar formation. The epidermis is the main structural support for skin and protects us from physical shock. For step 1, we need cells for the cell culture. The main groups of cells that can be used are autologous, which do not cause immune rejection and have low tumorigenicity since their DNA is not manipulated. On the other hand, we have allogenic, which are more likely to be rejected by the body because they are from another person and may only be used temporarily. Fibroblasts and keratinocytes are common cells used in products for wound and burn healing due to the functions previously mentioned. Now, the second step is crucial because autologous cells are isolated and expanded. This procedure can take up to 3 weeks. Cells need to be cultured in optimal conditions similar to a living organism. For example, autologous keratinocytes taken from a patient's skin biopsy can be cultured into a surface that supports cell growth and that can adhere properly. Fun fact, skin is the first organ to be tissue engineered. This began with the ability to culture and expand keratinocytes in 1975.
Hi, I'm Dr. One and I came to help with the case.
For step 3, cells from the cell culture are seeded into a scaffold structure. This is a temporary 3D structure, similar to the extracellular matrix to imitate the skin's anatomy in real conditions. So it guides the cell adhesion, growth, and differentiation to form the new skin. Now, the final step is to deliver all this to John. The main forms of delivery include dressing, 3D bioprinting, and cell spraying. These techniques are directly related to biotechnology, but will not be discussed in this session. Life is not perfect, guys. So, for example, although autologous cells are ideal for treatment, they can't be used on large scales because of the lack of donor sites and time. Fortunately, research continues and cell culture will continue being a fundamental step towards developing future treatment techniques.
Excellent work. You can party now.
Thank you, guys. Now you know the basics of cell culture.